Assalamu alaikum again, and let's continue with the carbohydrates series of lectures. So we finished last time the discussion of the organic chemistry part and information related to carbohydrates, in addition to some examples on monosaccharides, the most famous ones and important ones to us, like ribose, fructose, glucose, galactose, etc. Now we're going to talk about and give examples on some modifications that can happen to sugars and produces different types of molecules. Again, they're considered as sugars, and they're considered actually as modified sugars. Let's have some examples, starting with oxidation reactions can happen to sugars producing sugar acids. So oxidation can happen at different sites on a sugar structure. For example, on this the glucose can get oxidized through the OH group on carbon number 6 that is protruding, or that is excluded outside of the ring. So it can get oxidized into carboxylic acid, producing glucuronate or glucuronic acid. Glucuronic acid, once it donates its proton, it becomes glucuronate, as you can see in here. So oxidation can happen at carbon number 6. This is one example, and oxidation can happen also on carbon number 1. Okay, specifically when we talk about aldehyde sugars like glucose. In this situation, the produced sugar acid is called gluconate or gluconic acid. So oxidation on carbon number 1 produces gluconate or gluconic acid. On carbon number 6, glucuronic acid. So this again applies to galactose, for example. So it can produce a galactonate and galacturonate. Okay, so again, same idea applies. So let's see how this oxidation can happen. So definitely when we talk about oxidation, we need an oxidizing agent, and we may have a weak oxidizing agent or a strong oxidizing agent. So if the oxidizing agent is a weak one, which or where do you expect oxidation to happen, at carbon number 1 or carbon number 6? If you look at the structure of the glucose, carbon number 1 is the carbonyl carbon or the anomeric carbon, whereas carbon number 6 is an alcohol, or a carbon that is attached to a hydroxyl group or alcohol group. So if you remember from organic chemistry, oxidation of an aldehyde is just one step away from becoming a carboxylic acid. So it's just a one-step oxidation of an aldehyde to carboxylic acid, whereas the oxidation of OH group to carboxylic acid needs 2 steps: OH or alcohol to aldehyde, primary alcohol to aldehyde, and then aldehyde to carboxylic acid. So when we deal with a weak oxidizing agent, that means that the priority or oxidation is going to happen to carbon number 1, preferably on carbon number 6, producing gluconate rather than glucuronate. Okay, so how about the carbon number 6? This one is going to get oxidized whenever we have a strong oxidizing agent, and this would definitely oxidize carbon number 1 as well, producing a sugar acid with 2 oxidized sites: carbon number 1, carbon number 6. So what is this product that has oxidation happened on both sides, on carbon number 1 and carbon number 6? And in this example, this molecule is galactose. It's not glucose, so it is a galacteric acid with 2 sides oxidized. So how can we get oxidation just on carbon number 6 without oxidation on carbon number 1? In this case, we need enzymes. Okay, enzymes are going to specifically oxidize carbon number 6 to glucuronic acids, glucuronic, galacturonic, etc. producing these types of products. So to avoid the oxidation of carbon number 1, we need an enzyme that specifically oxidizes this carbon or the last carbon number 6 on glucose, galactose, or whatever type of sugar. Okay. How about the ketoses? Is oxidation going to happen for ketoses as well? Actually, this is not going to happen because, as you know, aldehydes can get oxidized, or aldoses can get oxidized, but ketoses cannot get oxidized directly, because this carbon in the ketoses is the carbonyl carbon is connected to 2 carbons, and that is hard to so it is hard to add more oxygen or reduce hydrogen content or reduce electrons, which means oxidation. Whereas in aldehydes or aldoses, due to the presence of this hydrogen, it's easy to remove it and replace it, or add oxygen, remove this hydrogen, or remove electrons and get this molecule oxidized. But there is a way that we can oxidize ketoses indirectly, where they can get converted into aldoses first, and then the aldoses can get oxidized. So how can we convert a ketose to an aldose? By a rearrangement of the bonds and electrons. So these electrons are going to move, and this oxygen here, the carbonyl, is going to become an OH group. Then the double bond is going to get transferred between the 2 carbons right here, so it becomes like an enediol, and then the electrons are going to be transferred and rearranged again to produce a double bond between carbon number 1 and the oxygen, removing this hydrogen. So it's like we transferred the carbonyl from position number 2 to position number 1, making the ketose an aldose, and so it can be oxidized this way. So let's talk about some biochemical tests that can be used to distinguish aldehydes generally from ketones, and probably you've discussed these in the organic chemistry course. The first one is Benedict's test, in which a reagent that contains copper, Cu+2, is going to be added to an aldehyde, like for example glucose as an aldose, so it can interact with it, oxidizing it, like you can see in here. So it becomes carboxylic acid on here, on this position, the easier one, and this would precipitate the copper as a brick red precipitate, indicating the presence of an aldehyde. If, for example, a ketone is added to this reagent, it's not going to interact, leaving the color of the reagent as it is, so it stays as a blue one rather than becoming red or brick red color. Other tests include a Tollens test, in which the Tollens reagent contains an Ag, الفضة. So once the sugar interacts with it, it gets oxidized into a lactone. Lactone, as you can see in here, this is the anomeric carbon, it becomes double bond O rather than an OH, and this is the oxygen that it has already in its structure. And this would precipitate this Ag as a mirror on the test tube walls، فبتصير زي المراية فعليا على الجدار، الوعاء، الفلاسك، التست تيوب، whatever. Let's have a clinical application on this oxidation reaction. For example, this structure here shows vitamin C. It's not a sugar again, but it has a lactone, or is a lactone, an unsaturated lactone, as you can see in here. So there is a ring with 1 oxygen, and the adjacent carbon, or the carbon next to it, has a double bond O. There, it's unsaturated lactone because of this extra double bond inside the ring. So it's not a sugar, but it's just an example on lactone, unsaturated lactone actually. So this is the ascorbic acid, or the vitamin C, that you all know that is important for uplifting or improving your immunity or immune system. So if, for example, whatever food that contains this ascorbic acid is exposed to air and gets oxidized, the vitamin C is oxidized by air, this would lead to loss of activity of this vitamin C, okay, because it's going to produce another molecule due to this oxidation. So the vitamin C is no more active, or is not active anymore. That's why when you lack, or when somebody is deprived of fresh food that contains vitamin C, there would be, or they would have, vitamin C deficiency that results in scurvy. And that's what happens with sailors, al-bahara. They spend a long time in sea, or sailing, so they would have food with them, but they won't have it fresh. That's why the vitamin C inside it is inactive, and they would get deficiencies of vitamin C. And this vitamin is important for immunity, and it's important for synthesizing collagen protein that is necessary for your gums, اللثة, if we're talking about dental aspects, and it's important also for skin and other tissues in your bodies. So because the synthesis of collagen that needs vitamin C is compromised, and the quality of this collagen is going to be reduced due to this deficiency, they would have some lacerations or some tears, بتصير تمزقات يعني, in the skin, in the gums, and in other tissues as well, due to this deficiency. It's not a common disease, but it's an example where vitamin C deficiency results in a disease. Another example on modified sugars are sugar alcohols, where the sugars like sorbose, for example, this is a ketose sugar with 6 carbons, or xylulose, which is a pentose or ketopentose, okay, as you can see in here. They can get reduced. So we go back in terms of reactions, and we reduce this ketone group into an alcohol, into a secondary alcohol. Remember, a ketone is produced by oxidation of secondary alcohol. Okay, so if we reduce this ketone, we're going to get back these alcohols. So the xylulose produces xylitol, the sorbose produces sorbitol, and these again have the sweet taste, so they can be used as sugars. Another example is also mannitol, etc., and they are used to sweeten some food products, specifically the sugar-free gum. If you read on it, on the ingredients list, you can find, or you may find, that it contains sorbitol, for example, or xylitol, or whatever type of reduced sugar. So they get reduced to sugar alcohols by or on their ketone group, the carbonyl of the ketone group. Other types of or examples of modified sugars or reduced sugars specifically is the 2-deoxyribose, or السكر خماسي منقوص الأكسجين, that is found in DNA structure. So it's originally derived of ribose, which is an aldopentose, as you can see in here, but the OH group on position, or on carbon number 2, is going to get removed and replaced with a hydrogen. So we lost this oxygen. That's why we call it deoxyribonucleic acid, اللي هو DNA. The ribose, on the other hand, is present in the RNA structure. Okay, so this is present in the RNA, and this one, the deoxyribose, is present in the DNA, and the deoxyribose is a reduced sugar by removing this OH group. Another example on sugar modifications is esterification of sugars and producing sugar esters. And this happens by adding phosphate groups, okay, as you can see in here. So this is a glucose molecule where we added a phosphate group through the OH group on carbon number 6, producing glucose 6-phosphate, or we can phosphorylation can happen on another site on carbon number 1 through the OH group on carbon number 1, producing glucose 1-phosphate. We call it esterification because it's very similar to the ester bond. So remember from organic chemistry that an ester has the following formula: R C double bond O O R. So it's R C double bond O O R. So it's just like an ester, but instead of carbon it's a phosphate. P double bond O O R. Okay, that's why we, or again here, P double bond O O R. That's why we call it phosphate ester. It's a phosphate ester, and this is a phosphate ester linkage. This is a phosphate ester linkage again, and this is a phosphate ester linkage. This is a phosphate acetal linkage because again this molecule follows the formula of an acetal. This is the carbon connected to one OR and one OP instead of OR, so it's a phosphoacetal. This one does not follow that formula, and it's just like an ester. That's why it's called a phosphate ester linkage. And the importance of these reactions is or can be seen during the studying of metabolism. For example, the phosphorylation on carbon number 6 when producing glucose 6-phosphate is important to mark a glucose molecule for degradation in the process or in the reactions of glycolysis. Glucose 1-phosphate is important for other series or other types of reactions in metabolism that you're going to get exposed inshallah in the next semester. Another example on modified sugars are the O-glycosides, or in which the sugars, like for example this glucose, can get bonded through the oxygen of the OH on carbon number 1 or the anomeric carbon to, for example, another sugar or another molecule like this alcohol molecule, forming this glycosidic bond. So the interaction between the anomeric carbon and alcohol group, whether it's part of an alcohol molecule or whether it's part of another sugar molecule, this is going to produce the glycosidic bond. Because the connection happened through this oxygen, so the produced or the resulting molecule is called O-glycoside. So this is the O-glycoside, or it's a disaccharide actually, a disaccharide, like as you can see in here, because the connection happened through this oxygen. This is important for producing the more complex sugars like disaccharides, oligosaccharides, polysaccharides, etc. And it's also important for glycosylation of proteins and glycoproteins, as it happens through the oxygen for O-linked glycosylation, and through nitrogen, as you can see in here, in N-linked glycosylation. Okay. The other type of modified sugars is the N-glycosides, where the sugars have some modifications in which a group with nitrogen is added to it, like an amino group, as you can see in here. Okay. Also, the sugars that are present in DNA and RNA, they are connected to nitrogenous bases through their nitrogens, or the nitrogenous bases nitrogens. That's why this connection makes them N-glycosides, or make the nucleotides of DNA or RNA as N-glycosides. These glycosides can be classified into two types: furanosides and pyranosides, depending whether the sugars that make up their structure are furans or related to furan structure, furanoses, or if they are pyranoses or having the 6-membered ring, as we've seen earlier. Okay. No matter the connection happened through a nitrogen, oxygen, etc., but they get classified depending on their ring structure. Another example on amino sugars, these modifications, for example, and replacements. For example, this glucose, and it can be galactose as well, where carbon number 2 get modified and the OH group is removed and replaced by an amino group. The amino group on position number 2 makes the structure of this sugar different, and now it's considered as the glucosamine or 2-glucosamine. Another modification, if you look at this same sugar with the amino group, the glucosamine, now having or forming an amide bond with a C double bond O, CH3. So this is a 2-carbon unit, that's why it's an acetyl group. It's attached through the nitrogen, we call it N-acetyl, and then 2-glucosamine. Okay, N-acetyl glucosamine, glucosamine, N-acetyl galactosamine are also present. By adding the amino group first, and for the acetyl glucosamine and acetyl glucosamine by acetylation next, or in the second step. Another example on modified sugars, the N-acetylneuraminate. This sugar, as you can see in here, is a modified sugar. It has this carboxyl right here in addition to the OH group. Additionally, it has an amino group on carbon number 4, and it has an acetyl group on the amide linkage, as you can see in here. So it's an amino sugar. The precursor of it is an amino sugar, neuraminic acid. Okay, so it's an N-acetylneuraminate. So we're adding this acetyl group to this structure of neuramic acid. The other name of it is sialic acid. This one is important as a residue or as a sugar that is present in the oligosaccharide chain of glycoproteins and glycolipids. So the glycoproteins and glycolipids have a group of sugars. It's not just one sugar or monosaccharide. They share, and specifically the glycolipids, they share the presence of this sialic acid, specifically at the terminal position. Let's now move to a little bit more complex sugars, which are disaccharides, in which we have two sugars or two monosaccharides connected together. If they are made of the same type of of the sugar, they're called, or they're considered as homo disaccharides. If they are of different sugars or two different sugars, they're considered as heterodisaccharides. The reaction that happens between them that we discussed earlier is an interaction between the first sugar through the anomeric carbon and the second sugar through another carbon that has an alcohol group on it. There are several questions that we're going to answer through this group of slides that discusses the disaccharide structure and characteristics. Okay. These disaccharides are synthesized by special enzymes called glycosyl transferases that transfer one sugar and attaches it to another sugar. Okay. Okay. There are several factors that can affect the structure and type of disaccharides that can be formed. First of all, we have two sugars. They can be of the same type or different types. They can be in a D configuration both, or an L configuration both, or a combination of D and L or L. We can have different carbons making the linkage between these sugars. So it can be between carbon number 1 and carbon number 4, carbon number 1 and carbon number 2, carbon number 1 and carbon number 6, etc. the order of these monomers or monosaccharides, which one is first, which one is the second, the orientation of the or configuration of the OH group on the anomeric carbon, whether it is upwards or downwards, alpha or beta, etc. So all these factors affect or make different types of disaccharides, as we can see in the following examples. Okay, so for example, the first one right here is the saccharose. Saccharose is the common table sugar. It's made of glucose and fructose. So this is glucose, look downwards right here, and this is the fructose, 2 carbons outside, and the connection happened through carbon number 1 on the glucose, the anomeric carbon, and carbon number 2 on the fructose. Okay, and you can tell easily which carbon is number 2 and which is carbon number 5. Carbon number 2 originally has an OH and the CH2OH, but carbon number 5 has just CH2OH. It doesn't have an OH group on it, so it has just a hydrogen. So this connection happened between the two anomerics, carbon number 1 and carbon number 2, producing this saccharose. The second example of disaccharides is the lactose. The lactose is made of galactose first and then glucose. Okay, so this is the galactose, the first one. Look, the OH is upwards, so it's a beta one, whereas the glucose is an alpha. Okay, they're connecting, and notice that the shape of this bond is like the N letter in English alphabet, and this is considered as a beta linkage because it happened between this beta sugar and this glucose. And remember that in glucose, the OH group on carbon number 4 is oriented downwards. That's why this linkage has to be with this form or with this shape, and it's considered as a beta 1,4 linkage. Lactose is the milk sugar. Okay, and the last one example right here is the maltose. Maltose is a homodisaccharide which is made of 2 glucose residues. The first glucose is an alpha one, as you can see in here. That's why the OH group is downwards. The OH group on number 4 is also directed downwards, and the linkage shape is like number 7 in Arabic. Okay, so, or V in English. So this V-shaped linkage is called alpha linkage. It happened between carbon number 1 and carbon number 4, producing alpha 1,4 linkage. Okay, an important thing to note that for monosaccharides, for example, like glucose, galactose, different examples, we've seen that they were able to get oxidized and get modified by oxidation. So this means that they are reducing sugars because they get oxidized and they reduce the oxidizing agent. Okay, and the only example that we discussed was in terms of or was discussed as a ketose was fructose, and we told you or I said to you that it can get converted to an aldose first, and then it can be oxidized. So all these monosaccharides can get oxidized and they're considered reducing. Now, how about these disaccharides? Are they still reducing or not? Let's look at the structure of these. Start first with the saccharose, for example. The position of oxidation, the easy one, we said, was the anomeric carbon, so carbon number 1 and carbon number 2 in case of fructose. Okay, and it has to, I know it has to become carbon number 1 by the isomerization, and then it can get oxidized. Notice that these carbons are connected via a linkage, so now they're unable to get oxidized. The OH groups now are not really available for oxidation. That's why this disaccharide saccharose is non-reducing. It cannot get oxidized. Whereas for lactose, for example, that is that has the galactose and glucose. The OH here was consumed in forming this bond, but the OH in the second one, or in the glucose, is still there, so it can get oxidized on this position, the easy position for oxidation, easier than position number 6, as you can see in here. That's why this is our reference in defining whether a sugar is oxidizing or whether it's a whether a sugar is a reducing sugar or not. So it still has an oxidative ability, so it can get oxidized. That's why lactose is a reducing sugar. Looking at the maltose, or the sugar of malt, سكر الشعير, that is present as well in Maltesers. Candy or chocolate balls. The maltose is made of 2 glucose residues, as you can see in here. It has this OH consumed in the reaction or in the bond, but the second OH, or the OH of the second residue, is still there, so it can get oxidized. That's why this sugar sorry, is a reducing sugar. Okay, so they're all reducing except saccharose, as well, and all the monosaccharides are actually reducing. So this is again to show you these sugars, saccharose made of glucose and fructose, and they're all made of these. They made of these 2 hexoses. It's made of these 2 hexoses. Lactose is made of glucose, and then is made of yes, glucose and galactose. The lactose is made of galactose first, and then glucose, again 2 hexoses. The maltose again is made of 2 hexoses, 2 glucose residues of the same type, and making this alpha linkage. Here the linkage is beta between galactose and glucose, and the linkage between glucose and fructose is a numeric 1-2 linkage. So we have different forms of disaccharides. We have an alpha 1,4 linkage that we've seen, for example, between these 2 glucose residues in the maltose. We're going to see other examples later on with alpha 1,6 linkage, as you can see in here, a glycosidic bond type. We've seen the beta linkage in lactose, the only one with a beta linkage as a disaccharide, 1,4 linkage, as you can see in here. There are also some other examples where we can get a glycosidic linkage between carbon number 1 and carbon number 1 of glucose, beta linkage. So we have different forms of disaccharides, different linkages depending on the, as I said before, on the what type of sugars we have, their alpha beta configuration, their order, which one is first, which one is the second, and some other factors that affect the structure or the formation of these sugars. Here again we have the saccharose, as you can see in here. This is the glucose, this is the fructose. They interact through the anomeric carbons, and as I said before, you can tell that this is carbon number 2 rather than number 5, because number 2 is going to have an OH as well as the CH2OH, whereas number 5 has only CH2OH and hydrogen and doesn't have an OH group here. A synthetic molecule, a synthetic molecule called sucralose was synthesized very close in terms of structure to saccharose. So this is the structure of saccharose, and this is the structure of sucralose, where this OH group was replaced with Cl. Here we have also Cl instead of OH, and here we have another Cl. So this molecule is still has a sweet taste and is considered as an artificial sweetener, محلي صناعي, just like that present in Splenda, or this brand of artificial sweeteners, in which sucralose is used actually as an artificial sweetener. but keep in mind that all other sugars that we've discussed earlier, as examples on monosaccharides or disaccharides, are actually naturally occurring compounds. These are artificial sweeteners. These are artificial compounds. The sucralose and other artificial sweeteners like aspartame, that is actually, in terms of structure, is a dipeptide, and saccharin, etc. Other types of artificial sweeteners. They have some debate, and there is a controversy about the use of these molecules as a replacement or as an alternative of natural sweeteners, especially that it becomes like a trend, especially between females and even males who are willing to lose their weight and not diabetic really. They're used mainly for diabetic patients, but now it becomes like a trend, and like between youth who can use it to make their sweets and cakes and whatever type of desserts instead of the saccharose or the common table sugar. But there are some reports in animals that these are harmful to health, but nothing is proved in human beings until now. You can search more for that and see whether there are any new reports or studies that investigated the effect of using these molecules in human beings. But you have to be careful if you're going to use them, because you never guarantee that this side effect that appeared in animals would not appear in humans. It may appear as well in humans. It may take a long time to appear. It may appear in the short run. That is unexpectable. Another problem or clinical application related to disaccharides is the lactose intolerance. In this example, you probably heard from many people that, and even you yourself, that you don't like milk anymore. You don't feel well after you drink a cup of milk, for example, and this problem may increase as you get older in age. And you probably hear that most people who are actually old having some problems with drinking milk. This actually became like the natural situation where most people cannot drink milk with no symptoms or with no problems after it. This is because of the lack of the enzyme, or the deficiency of an enzyme called lactase that is present in the intestine or protrudes from intestinal cells. So this enzyme is responsible for breaking the beta-1,4 linkage between the galactose and glucose in lactose, producing these monosaccharides that can be absorbed. So whenever we have this lactose accumulating because of the deficiency of the enzyme, and actually some textbooks say that it's a deficiency of the enzyme, so the amount of the enzyme is reduced. Others say that the enzyme is present but its activity is reduced. So anyways, there's less digestion of lactose in the intestine of these patients, so there would be no absorption of this disaccharide because it's considered as a large molecule in comparison to the composing or the ingredient monosaccharides. So this disaccharide accumulates in the intestine, and this would attract water molecules towards the intestine to reduce the concentration of lactose. Osmotic regulation, تنظيم الضغط الأسموزي. Okay, so the attraction of water molecules to reduce the concentration of lactose in the intestine would result in diarrhea. عشان هيك بصير في عندهم إيش؟ إسهال. Another symptom that they would have is bloating and gases. بصير في عندهم بقول لكم بصير عندهم نفخة أو غازات إلى آخره. Because of the following: this lactose is going to be used by the normal flora or the normal bacteria that is present in our intestines, and it enters the reactions and produces or the metabolic reactions and produces some side product of CO2, methane gas, etc. And this would result in bloating that is seen in these patients. So what's the solution in this situation? Just don't drink milk, because at this age, at an older age, patients do not really need milk that much. It's a source of calcium that can be obtained from other dairy products like yogurt, like cheese. So they don't have to drink milk to obtain calcium. For vitamin D, for example, they would not rely on milk to get vitamin D, because the amount that is needed daily of vitamin D needs or necessitate the consumption of like 10 liters of milk if you're going to obtain that amount of vitamin D, and that is not realistic, whether you have a problem with lactose or you don't. That's why just rely on getting exposed to sun to synthesize vitamin D rather than consuming it or obtaining it from milk. Okay. You may ask a question about the yogurt or cheese. Actually, the processing and the synthesis of these different dairy products is going to reduce this problem and reduces the lactose content inside. That's why they don't feel problems or symptoms after consuming cheese or yogurt, but they do feel that after consuming milk. Okay. Another solution, rather than stopping or stop drinking milk, the other solution is just by drinking milk that contains the enzyme, or drinking milk that does not contain lactose. Okay, and these some of them are present as commercial product that is sold in the market. Another problem related to disaccharides or to sugars is galactosemia. And in this problem, this is a genetic problem in which a galactose metabolizing enzyme, okay, is missing or is deficient due to a genetic mutation. This would result in accumulation of galactose, okay, so it does not get metabolized. So the accumulation of galactose inside the cells is going to activate the formation of a hydroxy sugar, galactitol, from galactose. Okay, this galactitol as hydroxy sugar or as an alcohol, sugar alcohol, cannot exit the cells. Okay, instead it would disturb the osmotic pressure inside the cell because the concentration of this solute is increasing. So what would happen is the attraction of water molecules and drawing these molecules towards the inside of the cell, resulting in swelling of these cells and damaging them at the end. Since we're talking about a genetic problem, so this problem is going to happen at an early age and would result in damaging very vital tissues like brain and for example the lens of the eye. So if it once it damages the brain, it results in mental retardation, and it's irreversible definitely, and it also results in cataracts أو إعتام عدسة العين اللي بنسميها بالعامية المية البيضاء. So this raffinose is an example on oligosaccharides actually, and it's made of 3 sugars: galactose, glucose, and fructose. The bond, as you can see, is different than what we've seen before. This is the galactose between carbon number 1 and carbon number 6 of glucose, and this is the glucose between carbon number 1 and carbon number 2 as we've seen in saccharose. Okay, so we have three sugars or three monosaccharides connected together in raffinose. This oligosaccharide is present in beans and vegetables, cabbage, Brussels, broccoli, asparagus, etc. يعني إجمالا كل الخضروات والحبوب طبعا سوري مش الحبوب، البقول اللي هي البينز، والخضروات اللي بنحكي احنا عنها بالعامية بتعمل نفخة يعني عند الشخص لما بتناولها. So why does this happen? Okay, اللي هو bloating. Why does bloating happen? Because human beings lack the enzyme alpha-galactosidase that is necessary for digesting this oligosaccharide and breaking it down. So it would again accumulate in this intestine, and it can be consumed by the intestinal bacteria, and fermenting these raffinose molecules, producing different types of products or side products like hydrogen, methane, other CO2 and other gases, resulting in bloating. Okay. The last example on oligosaccharides is using them as different types of drugs. These are synthesized molecules. They're not naturally occurring compounds, but they are, in terms of structures, they're actually oligosaccharides, such as some antibiotics like streptomycin and erythromycin. This is the structure of streptomycin and this is erythromycin. Notice these are the sugar molecules. These are the sugar molecules as part of their structures. They can also be used as a cancer chemotherapeutic agents like doxorubicin. This one right here, this is a sugar molecule inside it, modified sugar, as you can see. There's its nitrogen. And we can also see it in digoxin, the cardiovascular disease drug. You can see the sugar molecules as you can see in here, connected together and to other parts of the molecule. So with this, we're done with disaccharides and oligosaccharides, and in the next video, inshallah, we'll discuss polysaccharides.